Hi, my name is Casey Broker,
And I'm Michael Doraz.
and we will be talking about the distribution of large microplastics in Tampa Bay surface waters. This is underneath the Microplastics Monitoring Project here at Eckerd College with the guidance of Dr. Amy Sweda and Dr. Shannon Gowans. In recent years, humans have begun posing a unique threat to the marine environment. Our consumption of plastic products has led to the fragmentation of these products in Tampa Bay, and this becomes what we know as microplastics. And this is any plastic that is less than 5 millimeters. Seagrass beds are one of the primary habitats in Tampa Bay, and this is one of the first projects that is investigating microplastics distribution in this region. The objectives of the study are to describe the temporal and spatial distributions of microplastics, as well as investigating if microplastics are aggregating on floating dead seagrass. So on a bi-monthly basis, we collect water and seagrass samples from 7 different stations, which are pictured in Figure 1, using a 1,000 micron Newston net, which is figured in Figure 2. The samples were then stored in a cod end, and any excess seagrass was either added to the cod end or stored in a small bucket. The seagrass was then processed manually by scraping the surface with fingers, rinsing forceps and fingers into a 1,000 milliliter beaker. Seagrass was then dried and weighed, and the water samples were sieved, dyed, and photographed. These photos were then stitched together using Microsoft Ice and quantified using ImageJ. ANOVAs were run on the temporal and spatial data sets, and a linear regression was performed on a log transform data set to determine the relationship between seagrass abundance and microplastics concentrations.
The spatial distribution of microplastics at each of the 7 stations was observed as depicted in Figure 3. We also conducted an analysis of the temporal distribution of microplastics for each month from November 2017 to the present, as shown in Figure 4. We then determined if any correlation between the seagrass abundance and microplastic count existed, as demonstrated in Figure 5. Interestingly, the greatest mean microplastic count across the 20, 100, and 1,000 micron samples occurred during the month of July. However, for all data sets, no significance was found at the 95% confidence interval between independent variables. This alludes to the idea that more research is needed in the future to draw statistical conclusions. Our methodology for this research has been refined several times in the past 2 years in order to optimize results. If any conclusions can be drawn in the future, given the larger sample size, implications for graziers and browsers in seagrass beds, as well as the function of weather events on plastic concentrations can be explored. It is notable to mention that there may be a lag between weather events and microplastic concentrations and should be considered when analyzing future data sets. We would like to thank Eckerd College and the Tampa Bay Environmental Restoration Fund for funding and supporting our research. We would also like to thank all of the student researchers who assisted in data collection and analysis. And we look forward to what is next for our project.